Hello everybody. In this video I want to go over two techniques, two molecular biology techniques, PCR, which stands for polymerase chain reaction, and a method called Gibson assembly, which we use for cloning. This is an image of Taq DNA polymerase. This is a heat-stable DNA polymerase that's one of the key things that makes PCR work. So I'm going to talk about PCR first. And before I go over that, one of the key principles I want to introduce is what I'm referring to as the principle of hybridization. And what I'm referring to is the Watson-Crick base pairing between nucleic acids, which I think many of you are familiar with. And this is where A's pairs with T's and C's pairs with G's. So in an example of double-stranded DNA, and by the way, when we write out strands of DNA, get in the habit of always including the 5' end and the 3' end of DNA. So here's an example where you've got you got a strand of DNA, and then on the opposite strand, in the anti-parallel direction, so the 5' end is going to be down here, we have the other strand that is running the opposite direction. And what is holding these two strands together are the hydrogen bonds between the C's and the G's and the A's and the T's. And you guys are familiar with this from the structure of DNA. When we are representing these strands, we'll typically show them like this. Okay? And again, I've given you this note here of always get in the habit of labeling the 5' and the 3' ends. The reason I'm going over this is going to be really important for how we design primers for PCR. So here's a question for you guys to think about. What are factors that would affect the stability of a double-stranded DNA molecule? And I'm spilling the beans here. So one would be the identity of the match between two strands. So if you have two DNA strands and they are perfectly complementary to each other, that's going to increase the stability. If you have a mutation in there, it's going to make that double-strand DNA more likely to fall apart. The length of the DNA sequence is going to affect its stability. Longer sequences are going to require more energy to separate them. The percent GC content is going to be important. So remember, G's and C's have 3 hydrogen bonds that connect the bases, whereas A's and T's only have 2. So the more GCs you have, the higher percentage of GCs you have in a DNA sequence, the more stable that double-strand DNA will be. Temperature is going to be a factor, so that you can basically end up breaking hydrogen bonds by increasing the temperature. And it turns out that salt is also going to be important. So all of these factors are going to be relevant to PCR because at its base, PCR is based on two strands of DNA coming together. So there's a concept I want you guys to know which is called the melting temperature, which we call TM. And this is relevant for the design of PCR primers. So it is defined as the melting temperature of DNA is defined as the temperature at which 50% of DNA molecules are single-stranded, they're denatured. And this is what this might look like if you were to do this analysis. So imagine you were to put a strand of DNA into a reaction tube and gradually heat up the temperature and monitor when that sample became single-stranded. So at the very beginning, it would be all double-stranded here. And then as you increase the temperature, at some point, it's going to undergo this sigmoidal transition until all of the molecules will be single-stranded. So the TM is defined as the point at which 50% of the molecules are in their single-stranded state. This is a very critical parameter for designing PCR primers. We are going to be estimating what the TM of strands of DNA that we're going to use for our primers. And we're going to calculate a theoretical TM. There's some calculations that we could do. There's also web-based tools, which is what we are in fact going to do. So with that in mind, I want to go over the polymerase chain reaction, and I'm going to show you a PowerPoint from my colleague, Dr. Zanis. So PCR is a technique for amplifying a specific DNA sequence of interest. It consists of 3 steps that we do in repetition. There's a heating, a cooling, and then a replication step. And this brings out what we call this chain reaction, in which you exponentially produce DNA molecules. So, and the key to this is a heat-stable DNA polymerase. I showed you a Taq polymerase on the first slide, but now that we use other versions that have some benefits that are a little bit better, including their higher fidelity when they do the replication. So I want to go over the steps of PCR. Okay, so we're going to go over the steps of the polymerase chain reaction. And again, there's these 3 steps, which are going to be the denaturation step, the annealing step, and the extension step. And I'm going to go over those. So here are the things that you would end up having in a PCR reaction. You're going to need what's called a template DNA strand. So this is the DNA sequence that contains the sequence of DNA that you would like to amplify. You're going to need a pair of what are called primers, and you need 2 of them. These are going to be single-stranded pieces of DNA. We call them single-stranded oligonucleotides, and we'll go over why these are important in a second. You're going to need free nucleotides, those A, T, C's, and G's. These are the building blocks that are going to be required for DNA replication. And then you need a DNA polymerase. We need a Taq DNA polymerase. And what's unique about these polymerases is they are heat stable. So you can heat them to 95 degrees, and they don't denature, whereas many proteins do. And the reason that is because these were isolated from organisms that lived in thermal hot springs. They evolved to have heat-stable enzymes. Okay, so let's go through these steps. So I just talked about the Taq polymerase. This is a photo by my Uncle Jerry of one of these hot springs from which Thermus aquaticus, one of these heat-stable organisms, was isolated, and then the polymerase, the Taq polymerase, you can see Thermus aquaticus, was isolated from this organism. Okay, so here's the steps of PCR. The first thing you're going to do is you are going to heat up your DNA, all right? And we heat up to 95 degrees. And what that's going to do is it's going to separate the double-stranded DNA into single-stranded DNA, make sure all these primers are also single-stranded. And it's not going to break the backbone of DNA, so we're still going to keep the polymer intact, but it's going to break the double-stranded DNA into single strands, okay? So the first step, we do this at 95 degrees. Then you cool the DNA, and the temperature to which you cool is entirely defined by how you've designed your primers. So we're going to spend a lot of time thinking about the primers that we're going to design, and it's going to be for this step, the second step, which is called the annealing temperature, okay? So what happens during the annealing temperature is you're going to have a primer that you design is going to bind to one strand, another primer is going to bind to the other strand, they're going to recognize their sequence, and then you're going to warm up the reaction to 72 degrees, which is the optimal temperature for Taq polymerase. And then what Taq polymerase is going to do, it's going to bind to the 3' ends of those primers. So I didn't draw the ends on here, but this would be an exposed 3' end. And the polymerase is going to start incorporating nucleotides. And it would actually go all the way to the end of the sequence. It would keep on going. But what you can see is it's synthesized a new strand of DNA. Okay, so this would be 1 round of PCR. Okay, now we're going to go into a second round of PCR. And generally we do 25 of these rounds. So we're going to repeat these steps. So look at what our DNA templates are now. Our DNA templates include things that were just amplified in the previous round. Okay, so we're going to heat those strands. The single strand, we're going to have the single strands that are going to be produced. And then we're going to cool it down to our annealing temperature. And primers are going to anneal. So we have a primer that's going to be, you see there's 2 strands that are going to be recognized by the red primers, 2 by the blue primers. Then we're going to go down to our 72 degree step where the Taq polymerase is going to synthesize those strands. And now we've gone through 2 rounds of PCR. So what we've done is, so PCR, imagine we did this another round. Each of these DNA molecules would then serve as templates for the next round of PCR. And what happens with this is the PCR is going to be an exponential. So with each round, this is what you're going to observe. If you started out with 1 DNA molecule in round 1, after 1 round of PCR, you're going to have 2. After 2 rounds of PCR, you're going to have 4. We've gone through 2 rounds of PCR, right? Now, after 3 rounds of PCR, it's going to be 8, right? You can imagine that these would be duplicated to give you 8 molecules. And then after another round, it'd be 16, 32, and so on. And so it's exponential. So after 25 rounds of PCR, theoretically, if it's 100% efficient, you would have 2 to the 25 molecule amplification. What this means is you can start out with a very small amount of DNA and end up with a very large amount of DNA in the end. Okay. One thing, I'm going to step backwards on this. One thing I want to point out is notice the sequence that we've amplified here. Okay, I'm gonna step back to a previous one, which is going to be defined by what the blue primer is and where the red primer is. It's going to be this region. But we started out with a piece of DNA that was actually quite a bit bigger, right? We've got this long piece of DNA, and we ended up with a piece of DNA that was actually defined. The boundaries were defined by where these primers were. So you can start out with a large piece of DNA, but what you end up amplifying is going to be only within the primers that you design. Okay, one thing I'd like to point out is the format of DNA sequences that you get from most online sources. And there is now a very large repository of public databases that have various different sequences from lots of different organisms. The convention is to display the coding strand on the DNA. And so I think it's easiest to basically look at an example of this. And we talked about this, I think, previously, but numerous formats of DNA sequences are read by various programs. And the most commonly used is this thing that's called the FASTA format, which is having a greater than sign followed by annotation. And any text on this is not read by the programs, but after the carriage return, everything after that is. Okay, so that's the format that's the FASTA format. Now, an open reading frame is defined by having a start ATG, and then the ribosome is that's going to set the reading frame, and the ribosome, when it's actually reading a messenger RNA, is going to go 3 codons at a time until it hits a stop codon. So an open reading frame is defined by starting with an ATG and then having a stop codon, and that's going to be all that sequence that's in between. And so I point this out because if you're ever getting sequences from a database, this is what they're going to look like. And just bear in mind that we're only seeing one strand of what would be in a cell a double-stranded molecule, and it's usually the plus strand that we're looking at. Okay, so we just went over great aspects of PCR, right? The fact that you can get a huge amount of DNA. But there's another feature of PCR that is incredibly useful for molecular biologists. And we use this in a number of different ways, but in particular, we use it for cloning. And this is that when you're designing your primers, any extra information, meaning additional DNA nucleotides that are added to the 5' end of your primers will end up being incorporated into the final amplified PCR product. Okay? Here's an example of this. So this would be our template DNA, showing the 5' and 3' strands in black. And then remember, we always have to generate a pair of primers. So in this particular primers, I have my plus strand primer, which is going to be this blue arrow. Now keep in mind, when I'm first starting out the very first round of PCR, what I'm representing here by this blue arrow is what is going to be complementary to the DNA, the template DNA sequence. And that's very important. So when we are designing our primers, this is the first thing we pay attention to, is we need to pay attention to the melting temperature of our primers. And it's going to be only this sequence that's going to be complementary that we pay attention to for the melting temperature. Right? But on the 5' end, we can add additional DNA sequences that are here. And I'm going to show you an example where we do that for Gibson assembly, but we do this in a number of, you can imagine it being useful for a lot of different contexts. And then likewise, we can do the same thing with our minus strand primers. Okay, we have a region of the primer that is complementary to the DNA strand, and then we can add additional information. And so if you were to go through a typical PCR reaction, typically I do between 20 to 30 cycles, 25 would be typical. Your very final PCR product would be shown down here. And notice that the product now contains the information that was on the plus strand primer and the minus strand primer, including everything that was in the DNA that was in between. I normally have students go through and write out the first 3 cycles of a PCR reaction, starting with a single DNA strand, and convincing yourself that this is actually true. I'm telling you this, but until you actually go through the process, it's a little bit hard to visualize, I think. So this would be a very good exercise for you, would just be to start with one strand of DNA, and then pretend you're in a PCR machine. Heat it up to 95 degrees, those 2 strands come apart. Then you would cool down to your annealing temperature, the primers would anneal, then you would go to the extension temperature, and the polymerase is going to extend. Boom, you've gone through 1 cycle. Now do that another 2 times, right? You're going to end up with 8 total molecules, and you have to keep track of what all these additional sequences are. but it would show you that the final product would end up containing that extra information. Okay. Excuse me. Okay. So I want to move now to a technique that's called the Gibson method. So we will end up doing a PCR reaction and going over primer design in another as part of an exercise in lab. But I want to show you the other technique that we're going to use, and you need to understand this technique because it guides what you do for your primer design. So Gibson assembly is simply a method that stitches together two fragments of DNA that contain complementary overlaps. And that's going to be represented here. So what we've got is two strands of DNA. One would be my red strand, one would be my green strand, and what I'm representing here in black is that there is a region of overlap on the red strand and the green strand that these are going to be complementary sequences to each other. Okay, and there are three. So what you end up doing is you'd end up mixing together two DNA strands, and then in a single pot reaction, and what we do is 50 degrees for 15 minutes. We're going to include a mix that has three different enzyme activities, and this is the whole trick to the trick is designing overlaps and then this cocktail of three enzymes. So here's what happens. At 50 degrees, there's an enzyme. This is one of the three enzymes that's called an exonuclease. And it's a 5' to 3' exonuclease that was derived from a phage called T5. And what this is going to do is it's going to look at double-strand DNA, and it's going to chew back on the 5' ends in the 5' to 3' direction. So you can see that happened here is over on this end. We chewed back from the 5' end here, chewed back from the 5' end here, chewed back from the 5' end here, and chewed back from the 5' end here. Now notice what this does is it exposes single-stranded DNA here and single-stranded DNA here, and these two sequences are complementary to each other because we've designed them that way. So in the same reaction, those two fragments will now anneal to each other because they're complementary sequences. So here's where the other two enzymes come into play. You have a polymerase, and this is going to be a DNA polymerase. We typically use one that's called fusion. It's like Taq. And what it's going to do is it's going to add nucleotides on the 3' end of any nicks, any gaps here, using this other, using this guy as a template. And it's basically going to end up synthesizing a DNA strand until it gets to the adjacent strand that's there. At which point, a DNA ligase is going to seal the nick, and you end up with these two fragments that are stitched together. And that's how the Gibson assembly works. It was used to, so you can generate huge amounts of fragments. And this is an example that was used by, where it was actually first published back in 2010 by a guy named Daniel Gibson. And what they were doing was all these little red guys indicate fragments of DNA that are 60 base pairs, but they have overlapping fragments. And they did a series of reactions where they would end up assembling these guys using that strategy to give you these blue fragments. And those blue fragments were then assembled using this strategy to give you these green fragments. They had 3 fragments. And then they ended up using this strategy to take those 3 fragments and give you one really large chromosome. And they ended up using this to, this is going to be a 16.5 kb genome that they ended up assembling into, starting with these 60 base pair fragments. Okay, so it's incredibly powerful. Here's how we use this most often in the lab. And you can see that this is my version of writing stuff out, but the point is going to be pretty clear here. So this is the overview of cloning with Gibson strategy. And the goal is to take a plasmid of interest, which I'm showing here, and clone in your insert, this would be your gene of interest, into the plasmid. And I've given you a little arrow right here, all right? So remember what Gibson does is it stitches together the ends of 2 DNAs. So what that means is you have to have linear DNA. So what the first thing that's important is you need to generate a linear form of this plasmid. And if you see what happened, I've said linearize here. And if you go over here to the right, what I'm showing here is I've kind of written this thing where it's kind of still a circle, but it's open, all right? And we refer to that as a linear DNA because it's not a closed circle. So the key point here is we need to linearize the vector, in this case, P target. And there's 2 strategies by which we can do this. We can actually do a PCR of this backbone. So we could design primers that actually start going opposite directions from this point and would generate this entire fragment and go around the entire plasmid backbone to generate this fragment. And it seems that most people I'm talking with these days, this is their strategy for linearizing plasmid by PCR. The other way that you can do it, and it's totally valid, is by doing what's called a restriction enzyme digest. And there is an N there, I just wrote it poorly. And so a restriction enzymes are enzymes that cut DNA at a very specific site. And so what we would suppose here is there would be 1 or 2 restriction sites within the DNA sequence that would be recognized by restriction enzymes of interest that we would cut here. So imagine you can linearize your vector. So that's going to be one thing that's going to happen. The other thing you have to do is you have to generate your insert by PCR. And you would do that by designing primers that would amplify your insert, but also contain extra information that overlap with the vector, which I'm showing here, and I've highlighted in orange, right? And so really the key thing with the Gibson assembly is designing your PCR primers so you get amplification of your insert and so that they've got this overlap sequence with the vector. You do your PCR reaction, you linearize your vector either by PCR or by restriction digest. You isolate those 2 fragments, and then you end up mixing them together in a Gibson mix, and voila, it seals this insert into your plasmid backbone. And it works. It works very well. Okay, so that is, so we went over PCR, we went over Gibson. The other thing we didn't go into great detail is actually going through the process of designing the PCR primers, which is kind of the key thing for this. And I'll just point out that what's going to determine, one of the key factors that's going to determine the design of our primers is going to be that annealing temperature, that TM. So we're going to make sure that we've got a sequence of DNA that's specific to our insert that will match a certain TM. Then we're going to add some additional information, and we do a PCR reaction of our insert to generate this fragment. Okay, thanks guys. This is the end of this video for now.